Welcome everyone to our exploration of the fascinating field of molecular biology. This field delves deep into the very heart of life, examining the fundamental building blocks and processes that underpin the existence of all living organisms. The image on this slide depicts a stylized representation of a DNA molecule, the iconic double helix that contains the genetic code for life. The vibrant colors and glowing particles symbolize the dynamic and intricate nature of the molecular world, which we will dive into during this presentation. As we journey through this topic, we will uncover the remarkable complexity and elegance of the mechanisms that sustain life at the most fundamental level. Get ready to embark on an eye-opening exploration of the molecular foundations of the living world. Biomolecules are the fundamental building blocks of life, and they come in four main categories: proteins, carbohydrates, lipids, and nucleic acids. Proteins are diverse and perform a wide range of functions, from providing structural support to catalyzing chemical reactions. Carbohydrates serve as energy sources and structural components, and they also play vital roles in cell recognition and communication. Lipids provide energy storage, form cell membranes, and act as signaling molecules. Nucleic acids, such as DNA and RNA, store and transmit genetic information, directing the synthesis of proteins. Together, these biomolecules work in intricate ways to sustain and regulate the complex processes that define living organisms. DNA is the genetic material that carries the instructions for life. It consists of two long chains of nucleotides, twisted into a double helix. The sugar phosphate backbone forms the outside, with the bases facing inward. DNA carries the genetic code, determining the sequence of amino acids in proteins. This ultimately defines an organism's traits and characteristics. DNA replicates before cell division, ensuring each daughter cell receives a complete copy of the genetic information. This allows the genetic information to be passed on to future generations. RNA is the messenger of genetic information, carrying instructions from DNA to the ribosomes where proteins are synthesized. Messenger RNA, mRNA, transports the genetic code from the nucleus to the ribosomes. Transfer RNA, tRNA, delivers the amino acids to the ribosomes, matching them to the codons on the mRNA. Ribosomal RNA, rRNA, forms the core of the ribosomes, providing the structural framework for protein synthesis. Together, these three main types of RNA work in harmony to enable the flow of genetic information and the production of proteins in the cell. Protein synthesis is a fundamental process in all living organisms, converting the genetic information stored in DNA into functional proteins. The first step is transcription, where the DNA is transcribed into mRNA, which carries the genetic code to the ribosomes. In the second step, translation, the mRNA is read by the ribosomes, which use tRNA molecules to assemble a chain of amino acids, forming a polypeptide chain. Finally, the polypeptide chain folds into a three-dimensional structure, determined by the specific sequence of amino acids, resulting in a functional protein. This process is essential for the proper functioning of cells and the overall health and development of organisms. The slide provides an overview of the three main levels of gene expression regulation: transcriptional control, post-transcriptional control, and post-translational control. Transcriptional control focuses on the initiation of transcription, where proteins binding to specific DNA sequences can regulate gene expression. Post-transcriptional control involves the stability and translation of mRNA molecules, influencing the amount of protein produced. Post-translational control refers to the modifications that can be made to proteins after translation, affecting their activity, stability, and localization. The image on the slide is a stylized illustration of a DNA molecule with various regulatory elements, such as promoters, enhancers, and silencers, depicted as colorful shapes attached to the DNA, with arrows indicating the direction of gene expression. This visually represents the complexity of gene regulation. Biotechnology is a rapidly advancing field, with a wide range of applications that are transforming our world. One of the most promising areas is gene therapy, where we can replace faulty genes with healthy ones, offering hope for treating genetic disorders. Genetic engineering allows us to modify organisms to enhance desirable traits, benefiting agriculture, medicine, and industry. DNA fingerprinting is a powerful tool used in forensics and paternity testing, allowing us to identify individuals based on their unique DNA patterns. Drug discovery is another key application, where we can develop new drugs and therapies by targeting specific genes or proteins. These biotechnology advancements are driving progress in human health, agriculture, and environmental sustainability. The image on this slide is a thought-provoking illustration that captures the ethical dilemmas posed by advances in molecular biology. It depicts a DNA molecule entwined with ethical symbols like a scale of justice, a human heart, and a brain, highlighting the complex issues we must consider. The three key ethical considerations highlighted on this slide are privacy, safety, and equity. Privacy is a major concern, as genetic information raises the potential for discrimination. Safety is crucial, as modifying organisms can have unintended consequences that require careful risk assessment. Equity is also essential, ensuring that the benefits of biotechnology are accessible to all individuals and communities. These are critical ethical issues that we must grapple with as we continue to push the boundaries of molecular biology.